So serum sickness is the reaction when the antibodies that have been given in the passive immunization will trigger an antibody response against them since they are foreign. And on the picture here you see the time course, which is going to be determining if there is actually a serum sickness developing. So let's say we are treating botulism with horse serum. And once we inject the horse serum, there are no anti-horse antibodies present, but they start after 1 to 2 weeks to form. But by this time, normally the antibodies from the horse serum should get cleared from the system. So there is a balance point where you have enough antibodies left once your own production of the antibodies has started. So the serum sickness is a reaction which is similar to an allergy. And it can also be a reaction against other proteins in the serum, not only the antibodies. They form then immune complexes with the antibodies that we produce ourselves, what is given. And these immune complexes will trigger the complement activation of the complement component 3, so C3. And these complexes will precipitate on the inside of the vessels. And in particular in the kidney is this precipitation is common, and these precipitates can lead then to that the epithelial wall in the kidney gets damaged by the complement. And the term for this is leukocytoclastic vasculitis. And it also results in hypocomplementemia, where the C3 levels in serum are very low. The symptoms of the serum sickness can be kidney related, but also rashes and fever and joint pain. Now we will move into the core of this lecture, which is the active immunization using vaccines. In order to do this, I would like to talk to you a little bit about the adaptive immune system. I know that you have heard about it and that you have lectures about it, but I would like to give you a briefing again and go over the parts which are really important. So, T-cell priming by activated dendritic cells, and how the dendritic cells are activated. We have been nosing on this before. This is the TLR activation of dendritic cells. But we will also talk a little bit about T-cell priming and the physiology of T-cell priming. We will also talk about follicular T helper cells, which are driving the B-cell responses, which are very important for vaccine development. So here goes a repetition of adaptive immune responses. So also now a quick review of the activation of the TLRs and the consequence of this activation. The TLRs are the most common way of activation of dendritic cells. And one reason for that is that the TLR induction, signaling induction, is leading to IL-12 production. And IL-12 production is very desirable when it comes to vaccination. So TLR ligation and activation via MYD88 will cause the upregulation of cytokines which are regulated by NF-kappa B. These cytokines will cause inflammation and also to some degree tissue damage. The chemokines CCL2 and CXCL8 will cause infiltration of other cells into the area, and this is also very important for the vaccination. What we will come into next is the upregulation of the co-stimulatory molecules. While dendritic cells which do not have co-stimulatory molecule is very poor at priming a T-cell response, the co-stimulatory molecules are absolutely required for efficient T-cell priming. They can be upregulated in many different ways, but the TLR signaling causes a very robust and reliable activation of the co-stimulation. In general, a rule of thumb is that the TLRs that are extracellular and on the surface, they will recognize bacterial products. The TLRs found inside of the endosome will recognize viral nucleic acid, but also to some degree bacteric acids of bacteria or remains of bacteria taken up into the endosome. Since all of the TLRs signal via their tear domain, they signal in a similar manner. The outcome of the different signals from the different TLR on the dendritic cell is not very big. So most of the TLRs will end up affecting the dendritic cell in the same way. What will differentiate the maturation of the dendritic cell is other signals which come in before or at the same time. For example, signaling via TLR and the CD40 receptor on the surface of dendritic cells, which can be activated by an anti-CD40 antibody, will cause an additional stimulation to the dendritic cell, which cannot be achieved from either the CD40 alone or the TLR alone. So also an addition here is that activation simultaneously via MYD88 and the TRIF pathway, which is activated by TLR3 and TLR4, gives an enhanced signal as compared to only activating the MYD88 or only activating the TRIF via TLR3. Therefore, the TLR4 is a very potent form of dendritic cell stimulation, and this is used in vitro. However, in vivo, the TLR4 activation using LPS is very dangerous, since you can have a very strong reaction. We will later come back to a slide where we talk about how the different adjuvants activate the different TLRs. But now we need to talk about the effect on the dendritic cell by the TLR activation and how this affects the T cell priming. The priming of a T cell response by a dendritic cell requires 3 signals to work optimally. The first signal is the MHC class 1 molecule or class 2, which is then binding to the T cell receptor, which is specific for the peptide in the grove of the MHC. But binding to the T cell receptor is not sufficient in itself. Without co-stimulation, this will lead to proliferation, but no killing and no cytokine production by the T cell. In order to prime a T cell properly, you need the same signal, and that is the co-stimulation. Co-stimulation occurs mainly via the CD28 molecule, which recognizes the B7 molecules on the surface of the antigen-presenting cell. The B7 molecule, also known as CD80, CD86, ligate to CD28 and sends in the co-stimulation signal into the T cell, as does the CD40 ligating to the CD40 ligand. The third signal is the cytokines produced by the dendritic cells. Optimally, for a cytotoxic response, IL-12 should be produced by the dendritic cell, and IL-12 is induced by danger signals, and particularly signals via the TLR receptors. The priming of a T cell, or the induction of a response in an already primed T cell, is also subject to inhibition. CTLA4 is binding the B7 molecule CD80, CD86, and therefore prevent them from binding to the CD28, and they do not then give any co-stimulation. PD1 is a molecule expressed on T cells and also on antigen-presenting cells, and this molecule signals into the T cells to prevent activation. It's also an activation marker, and it's upregulated on T cells after they have been activated. PDL1, expressed on the antigen-presenting cells, will induce signaling through the PD1, and the density of PDL on the dendritic cell is a measure of how inhibitory this dendritic cell is. Antigen presentation is initiated in the tissue, where dendritic cells are taking up antigen get activated and then go to the secondary lymphoid organs, the lymph nodes. Inside of the lymph node, the dendritic cell will encounter naive CD4 and CD8 T cells. The CD28 on the CD8 T cell will bind to the CD80, CD86, the B7 molecules on the surface of the dendritic cells. And this stimulation, together with cytokines released from the dendritic cell, will cause the T cells that recognize antigens on the surface of the dendritic cell to proliferate. Once the CD8 T cell has proliferated, it is becoming an effector T cell, which is capable of killing, or a cytotoxic T lymphocyte. The cytotoxic T lymphocyte effector cells are then exiting into circulation. Out in the circulation, they will encounter their targets, which present the same molecules on MHC class 1 in the body, and thereafter they will kill their targets through different measures, through death receptors or through the granzyme pathway. The T cell priming is initiated by antigen-presenting cells in the periphery taking up the antigen and then moving to the lymph nodes, the secondary lymphoid organs. There, the dendritic cell will encounter the naive CD8 T cells. The CD8 T cells express CD28, which is a co-stimulatory molecule on their surface. This binds to the B7 molecules, which are CD80 and CD86. Once the T cell has recognized its specific peptide on the surface of the antigen-presenting cell, the CD8 T cells are differentiating into effector cells and divide. The effector cells go out in the circulation where they find other cells presenting the peptide they recognize. If the priming has been successful, this results in the killing of the target cell. That's why they call the cytotoxic T lymphocytes. So in the active immunization, we need to induce a T cell response. And here we need to induce first the innate immunity by antigen and non-dangerous agents called adjuvants. And we need to, after this, induce the adaptive immune response against the antigen. And this will lead to the formation of memory cells and antibodies, which are class switched. So this means antibodies produced from plasma cells of the type of IgG. So if we go through the picture here step by step, the first step here is the recognition of the antigen priming naive CD4 T cells and naive CD8 T cells. The CD4 T cells will produce cytokines which will help both the CD8 T cells and the CD4 T cells themselves to proliferate. So I drives proliferation, and after proliferation the selection is going to happen so that the high affinity T cells will survive. This is called differentiation. And once the cells are fully differentiated, and for the effector CD4 T cells, that means that they can activate macrophages, they can activate B cells, which we'll come back to, and other cells in the inflammation. The differentiation is also directing a small part of the CD4 T cells into the memory pool of T cells. And the same thing is true for the effector cells. The CD8 effector cells will perform the direct killing of infected cells. They will also provide cytokines for macrophage activation, which will take care of the remains of the cells which have been killed by the CD8 T cells. And the same here, a small pool of memory CD8 T cells will form from a certain precursors at a certain state of development, and these memory T cells will only mature once all of the antigen is gone. Up until the point when all the antigen has disappeared from the system, the memory T cells will still have effector phenotype. So from this comes that the vaccination also needs to clear out the antigen at some point. You cannot have a very prolonged antigen production because this may impair memory formation.